Hello everyone, and thank you for being here. My name is Orrit, and today I will present part of my master thesis where I explore how cultural keystone species can help reveal patterns of biocultural vulnerability at global scale. To guide the presentation, I'll first introduce the conceptual framework, then explain the methodology, present the main findings, and finally discuss their implication for biocultural conservation. Turning now to the introduction. The global context of this research remains in the fact that despite decades of conservation efforts, biodiversity loss continues to accelerate globally. And at the same time, cultural and linguistic diversity are also declining. These two processes are deeply interconnected. Humans and ecosystems have co-evolved through long-term relationships where many indigenous and local communities actively maintain biodiversity through stewardship practices. The global change is disrupting this club nature cultural systems, which had led to growing interest in rethinking conservation through biocultural approaches. These approaches understand conservation as the joint maintenance of biological and cultural diversity with the dynamic socio-ecological systems. And one way to understand these linked dynamics is through the concept of cultural keystone species. In this context, Garibaldi and Turner proposed the concept, the cultural keystone species, around 20 years ago. And these are species that play a fundamental role in the cultural identity of people, not only through material uses, but also through knowledge, practices, and meanings. Here, for example, the sweetgrass is a well-known cultural keystone species for Wabanaki people in North America. We further add that, to this definition, that cultural keystone species are also embedded in the reciprocal relationships. They provide food, materials, health, and meaning, while being actively maintained through cultural root management or stewardship practices that foster their persistence across generations. Because they connect ecological and cultural systems, cultural keystone species offer a useful lens to study conservation beyond species alone. However, despite growing attention to this concept, few studies systematically combine biodiversity and cultural indicators to assess the cultural keystone species vulnerability. The main objective of this study is to assess the vulnerability of documented cultural keystone species at the global scale. To do so, we focused on three specific objectives. First, to characterize documented cultural keystone species taxonomically, culturally, and geographically. Second, to assess their biological, cultural, and biocultural vulnerability. And third, to explore temporal trends across these dimensions. To address these questions, we constructed a global database of documented cultural keystone species from the specific literature. With this systematic literature review, we used the exact term of cultural keystone species in an English language peer-reviewed publication indexed in Google Scholar. After screening and filtering, the review resulted in 308 publications reporting 1,170 cultural keystone species cases. From each study, we extracted information linking a biological taxon with a sociocultural group. And for each record, we compiled the taxonomic, geographic, biological, and cultural information. To determine the biological status, we obtained the categories from the International Union for Conservation of Nature, Red List, or the IUCN, while the cultural status was approximated through language vitality data from Ethnologue database, using language as a proxy for cultural continuity and ecological knowledge transmission. The temporal trends were determined assessing the repeated status assessment of both dimension and classifying the direction. And after compiling these records, we standardized and filtered the dataset for the analysis. To avoid the redundancy, duplicated records were removed from the original dataset, resulting in 938 unique documented cultural keystone species. From these, genus-only records were excluded, leaving 809 species-level cultural keystone species corresponding to 365 species and 349 languages, as we can see in this figure. Using the final dataset, we analyzed the taxonomic and geographic patterns, relationship between biological and cultural status, the biocultural vulnerability, and the temporal trends using programs such as RStudio and Excel. Turning now to the results, I'll begin with the taxonomic and cultural characterization of documented cultural keystone species. At the biological level, plants clearly dominate the documented cultural keystone species, representing around two-thirds of cases, while animals account for about one-third. Fungi have a marginal presence. Most plants belong to the class Magnolopsida, the 71%, while the most represented class of animals is Mammalia, the 32%, and only two fungi belong to the same class of Agaromycetes. At the cultural level, there is fairly equitable representation between linguistic families, with Austronesian being the most represented, followed by Australian and Algic. Beyond taxonomy, this dataset also reveals strong geographic patterns. The geographic distribution of documented cultural keystone species is highly uneven. More than half of the documented cases are concentrated in North America, and three countries, the United States, Canada, and Australia, account for nearly the 60% of the total, as we can see in the map here. At the subnational scale, the concentration of the North America Pacific coast stands out in both the United States and Canada. That is the dark blue there. This confirms the strong special and research bias in the current literature. After examining where documented cultural keystone species occur, we then explore their vulnerability patterns. Looking at the vulnerability patterns, we can see that most cultural keystone species are not biologically threatened. However, around the 60% are associated with groups whose languages are endangered. In fact, over one-third of documented cultural keystone species are biologically secure but culturally pressured. This means that many species remain ecologically stable while the cultural systems that sustain them are under threat. However, the picture changes considerably when biological and cultural dimensions are analyzed together. When cultural and biological dimensions are combined, as we can see in the left graphic, more than half of documented cultural keystone species become bioculturally vulnerable. This pattern is especially strong in animals, where almost 70% fall into this category when we do the analysis separated by kingdom. Importantly, this vulnerability remains largely invisible when the biological and cultural dimensions are analyzed separately, as we can see in the right graphic. In addition to these vulnerability patterns, we also explore how biological and cultural dimensions change over time. Temporal trends could only be assessed for a subset of cases. Biological status showed more frequent changes over time, while cultural status tended to change more slowly. This likely reflects the different process governing each dimensions. Species conservation status can respond relatively quickly to management or environmental pressures, where cultural systems depend on longer-term processes such as governance, education, and international transmission. Despite these differences, biological and cultural trends were still significantly associated, suggesting that these dynamics are not independent. These findings raised several discussion points. First, that the cultural keystone species dataset reveals important research and documentation bias. The predominance of plants likely reflects the ethnobotanical origins of the concepts, while the strong concentration in North America is linked both to the regions of the framework and to the English language focus on the literature review. In contrast, regions such as Africa, South America, and Europe remain underrepresented. This does not imply an absence of cultural relationships, but rather an even application of this framework. A clear example of this regional concentration is the case of the salmon in the Pacific Northwest. In our dataset, the salmons appear 71 mentions associated with multiple indigenous communities along the Pacific coast. Beyond these biases, the results also highlight an important conceptual pattern regarding the drivers of vulnerability. The second key finding is that bicultural vulnerability is often driven more by cultural pressure than by biological extinction risk. Language plays a fundamental role in transmission of indigenous and local ecological knowledge, while linguistic vitality weakens. Stewardship practice and reciprocal relationships with the species may also decline. This is the case of American bisons that illustrates this clearly, since the historical collapse had profound cultural consequences for many indigenous communities. Today, bison restoration is often framed not only as ecological recovery, but also as cultural revitalization. A third important point that concerns the temporal mismatch between biological and cultural recovery, as biological recovery does not necessarily ensure cultural recovery. Biological and cultural systems operate at different temporal and institutional scales, which can produce unsynchronized trajectories. The conservation status of a species can respond relatively quickly to management measures, regulations, or extractive pressures, and is supported by standardized monitoring systems such as IUCN and Red List. In contrast, linguistic vitality depends on long-term institutional and intergenerational process, such as the governance, education, and cultural transmissions, and often lacks such systematic monitoring systems. This asymmetry helps to explain why biological and cultural trajectories do not always evolve in synchronized manner. An illustrative example of this case is the Driobalanopus aromatica, species of cultural significance developed in different communities in Borneo, which improved its conservation status in recent decades, while associated language moved from stable classifications to threatened categories. This highlights that biocultural vulnerability is dynamic and cannot be understood through ecological indicators alone, and that conservation outcomes may diverge across dimensions. Before concluding, I would like to briefly acknowledge some limitations of the study. This study adopts a global synthesis based on the best available standard data. Therefore, the results should be interpreted as exploratory and aimed at identifying general patterns, bias, and knowledge gaps. Several limitations should be acknowledged, including the use of language vitality as a proxy for cultural status, which, although it is one of the few comparable global indicators, does not capture the complexity of cultural systems. Also, the geographic and taxonomic bias of available literature, which can leave out other research in other language or concepts that are not strictly cultural keystone species. And also the tensions between local and taxonomies and scientific classification. These limitations highlight the need for more contextualized indicators and stronger collaboration with local communities. But despite this, several broader conclusions can still be drawn from this analysis. To conclude, this study shows that cultural keystone species can be operationalized globally by integrating biological conservation status with linguistic vitality, but with some important research gaps should be addressed. Our approach reveals forms of biocultural vulnerability that remain hidden in purely ecological assessments. And more broadly, these findings reinforce the need for conservation approaches that recognize biodiversity and cultural diversity as deeply interconnected, and also suggest conservation policies focus exclusively on species or ecosystems may overlook critical cultural dimensions of biodiversity stewardship. We think that effective conservation requires protecting not only species and ecosystems, but also the cultural systems through which they are maintained. This is all the exposition. Thank you very much for your attention, and I'll be happy to take any questions. Thank you.